All right, Madeline.
It's a way of, like, bacteria cells, like a way of them being able to count and, like, sense of, like, how many other ones are around them, or like what species are around them. And it's beneficial so that they can assemble and, work on, like, jobs together, or, like, the path, like, how it relates, like, it takes over a host, so.
Okay, so we're going right into number 2, the second one here. Absolutely. So when we say that they have some kind of response based upon how many of them are present, what are they actually sensing to know the density or how many of the bacteria are present? So if this is a bacteria, this one single bacteria. What does it do, Scott?
It's just that they're secreting a molecule, and if there's enough of them, there's enough molecule, it will bond with a second bacteria and trigger a response.
That's correct. So it's releasing a molecule, and when there's just one of them, she talked about how that molecule will diffuse away and it won't really have an effect. Remember that the bacteria can actually communicate with itself or with other members of the same species, and so you have, and how it does that is by these receptors in the membrane. So it can communicate with itself or other members, and the reason why is because it has similar, the same receptors in it. And so when that diffuses away, when there's just a few bacteria, then there doesn't, no change happens. But what happens is either these signal molecules allow, as they diffuse away, this bacteria can divide. That's what bacteria do. You can have one bacteria in the matter of hours. You can have or 24 hours, you get millions of them stemming from that one bacteria. And so then as you get more and more of these bacteria, and all of these guys are secreting this signal molecule gets in high concentration, and then these signal molecules can bind to these receptors on the membrane, and all of that happens at once, and so then you get one big response, like with the Vibrio fischeri bacteria, where they all, she said that, when they were in low concentration, even in a flask in the lab, they did not emit any light, but when they were in high concentration, they did, and that is because of the signal molecules. So why is this beneficial to the bacteria? If the one bacteria, let's go with just that Vibrio fischeri. If you have one bacteria, let's say it did work and it emits bioluminescence, is any other organism going to really be able to see that? Can we see bacteria? Bacteria, even when we magnify it underneath the microscope, it looks like a little dot. All right, and so in real life we wouldn't be able to see, other organisms wouldn't be able to see that. So therefore, since they're so small, to elicit some kind of response or to do something in the larger environment, it takes lots of these bacteria, and so therefore it'd be very wasted for a single bacteria to make a protein and that will allow it to bioluminesce. And so what happens is they wait till there's many of them, have a high density, and then do it all together, which then they have more an effect on real life. So what happens is when this actually binds to it, when this binds here, I was talking here, what does that cause to happen? It causes a change to occur in the cell. A lot of times genes are turned on, which means then that they're able to make a protein. So in this case here, the gene to make the protein is turned on, and then the protein is what causes the bioluminescence. And so therefore, these genes are all the time in the bacteria, but just aren't turned on, and they have to have that signal molecule in order to do that. And so therefore, that's why all those genes would be turned on simultaneously. All those bacteria would make the protein simultaneously, and the bioluminescence would happen simultaneously as a group. So how does this relate to bacteria that happen to be a pathogen? So the Vibrio fischeri, the bioluminescence one, wasn't a pathogen. It helps, does bioluminescence and lives in like mutualistic relationship with what other organism? Is this one? The Vibrio fish, right? The squid, right? Which I think is amazing how the squid, it talks about, before I get into the next question, that the squid, it talked about how it has this living inside of it, and then it said the squid, according to its circadian rhythm, we'll talk about that more later on in the course, but circadian rhythm is a natural 24-hour cycle that living organisms do things by. And so oftentimes that can be triggered by light. All right, so it talked about in the daytime, the squid sleeps because it's nocturnal, and so before it actually goes underneath the sand, it pumps out 90% of this bacteria out of its body, and then it goes underneath the sand and it sleeps during the day. While it's sleeping, the bacteria that's left start dividing. So they start dividing. So now when the squid wakes up and it's nighttime, now you've got a bunch of bacteria in it, and guess what? It has enough density to trigger them, the genes to be turned on, and then they bioluminesce at night. And the squid, what was cool about the squid with this bioluminescence? How did it use? How was that helpful for the squid?
It eliminated its shadow.
That's right, it eliminated its shadow and talked about how it has a shutter. So the moonlight, where the light hits it, it adjusts the shutter so that the bioluminescence kind of counteracted its shadow where the moonlight was hitting it, and so therefore there was no shadow on the ground, and so therefore there would be no, way that predators would be able to find it. So that was kind of cool. With pathogenicity, how does this relate quorum sensing to, let's say, a bacteria infecting us? What do you think? Jacob?
This relates to like how bacteria inside us, they'll communicate, and when their numbers are high enough, they'll be able to make us sick.
Yes, absolutely, because bacteria often produce a chemical or some kind of substance that's toxic to us to make us sick. One bacteria, she talked about us being huge compared to this one little bacteria. So one little bacteria by itself and working by itself and producing this molecule, this toxic molecule to us, probably not going to have an effect on us as trillion cells big. And so therefore, it'd be a waste of that bacteria to try and do that. So therefore, the bacteria does quorum sensing, where it divides and it waits till there's many of the bacteria, and then all of that elicits a response altogether, and so therefore they have an effect that way. Intra versus interspecies communication. This one that I just talked about here, let's say that these guys are the same species. Let's say these are species A of the bacteria. Is this intra or inter species communication? What do you think, Madeline? This is intra. Intra is the same species. It means within, so within the species. Interspecies, inter means between, so that would be communicating between different species. And so this would be intra, and then at the same point, so bacteria have very specific molecules that they use to communicate with those of the same species, but they also have more generic molecules as well. So bacteria can produce generic molecules that can communicate with, let's say, species B here. And so species B and species A can quote-unquote talk by these other molecules, and then the organisms of the same species can also talk without species B being in on that conversation. And so there's two different ways in which species can communicate. Why can't, for the next one, why can't species B be in on the conversation between the ones with between species A? Megan?
It doesn't have the receptors that accept the binding.
Yeah, it doesn't have the right receptors. It all has to do with the proteins embedded in here. So species B has these kinds of receptors, and they might have their own receptors to communicate with other species B organisms, but what species B does not have is the receptors in the membrane that the triangles could bind to. And so therefore, these guys can talk amongst themselves without these guys quote unquote hearing, but they can also talk to them if they want to. All right, and so therefore generic versus the same. And lastly, how does this relate to antibiotics? So what did they say that they're doing research on? Madeline.
Like, it almost sounded to me like they were gonna like clog the receptors so that they can stop like making counts and being able to know where.
Yeah, they're trying to make molecules that would fit in the same receptor here. So if you have a molecule that would fit in this receptor, as Madeline used the word clog, it would clog the receptors or block them. So then the signal molecules, the triangles, would never be able to bind, and therefore the bacteria would never get to a point where they say, oh, let's elicit a response. And so therefore the bacteria, we would never have side effects from that because they would never think that they were dense enough to launch an attack. What that then does is gives your own immune system time to attack these bacteria and kill them naturally, all right, without the bacteria actually producing something that's going to kill you. Does that make sense? Okay, and this way, it doesn't kill the bacteria. We kill all the bacteria naturally, and it won't play into antibiotic resistance, all right, so some of you guys turned in your papers up here. You did not, let's put it up there.